Welcome to video series on protein metabolism. This first video is on digestion of proteins. Dietary proteins on cooking, they get denatured and are easily made digestible. Now, these denatured proteins are acted upon by pepsin, which is present in inactive form as zymogen, pepsinogen. Now, pepsinogen is activated by hydrochloric acid, and this pepsin is active at a pH of 1 to 2, and this degrades proteins to proteoses. This is taking place in adults. In infants, the milk protein, that is casein, is acted upon by renin, which converts casein to paracaseinate, and subsequently paracaseinate gets broken down to peptides. Proteoses enter into small intestine, that is duodenum. Now the pancreas releases the digestive enzymes under the stimulation by cholecystokinin, and this hormone stimulates the release of all the digestive enzymes of proteins. Further, enterokinase, which is the enzyme present at the brushport of the duodenum, activates conversion of trypsinogen to trypsin. All the digestive enzymes are present as inactive zymogen, mainly to prevent the autolysis of the cell. Trypsinogen is converted to trypsin by enterokinase. Now, trypsin in turn converts all other inactive enzymes to its active form. For example, chymotrypsin gets converted to chymotrypsin, proelastase gets converted to elastase. Trypsin, chymotrypsin, and elastase are endopeptidase, the enzymes acting at the center part of the protein molecule, whereas the procarboxypeptidase and the proaminopeptidase, which gets converted to carboxypeptidase and aminopeptidase, these are exopeptidase acting at the extreme end of the extreme amino terminal end or the carboxy terminal end of the protein. So in the duodenum, proteoses get converted to peptides. It can be a formation, they can end up with the formation of a dipeptide, tripeptide, or oligonucleotide. Peptides are broken down to amino acids by peptidase enzyme, and once these amino acids are formed in the intestinal lumen, they are absorbed into the general circulation by 5 different transport mechanisms. We have different transport mechanism for acidic, basic, amino acid, beta-alanine, and neutral amino acids. For acidic amino acids, mainly for aspartic and glutamic acid, we have a separate transport mechanism, and all these transport mechanisms utilize active transport mechanism, and they are dependent on sodium. Their sodium dependent active transport facilitates the transportation of different amino acids from the lumen into the general circulation. Now, for the basic amino acids, arginine and lysine, a separate transport mechanism is there. Glycine and amino acid proline, we have a separate transport mechanism. Next, there is one more transport mechanism for beta-alanine, and for the neutral amino acids, we have a separate transport mechanism, and the cycle involved in transportation of neutral amino acids, valine, leucine, isoleucine, alanine, methionine, phenylalanine, is known as Mr. cycle. So the Mr. cycle is as follows. The neutral amino acids combine with reduced glutathione. So glutathione is a tripeptide made up of gamma glutamate, cysteine, and glycine. Now, so the glutamyl group is transferred to amino acid, and they become glutamyl amino acids. Now, the glutamate get converted to oxoproline, releasing the amino acids which enter into the blood circulation. Oxoprolinase acts on oxoproline and converts it back to glutamate. Now this combines with cysteine, catalyzed when the enzyme synthetase requiring energy from ATP gets converted to gamma glutamyl cysteine. Then subsequently glycine is added and reduced glutathione is resynthesized back for further transport of neutral amino acids. Now, so this is the overview of the digestion and absorption of proteins. Just to summarize, dietary proteins on cooking gets denatured and are easily digestible. These are acted upon by pepsin, which is present in inactive form pepsinogen activated by HCl. So pepsin at a pH of 1 to 2 converts degrades protein to proteoses. Proteoses are acted upon by pancreatic enzymes. For example, trymosin. All these pancreatic enzymes are present as inactive zymogens. Trypsinogen is activated to trypsin by enterokinase enzyme, and trypsin further activates all other digestive enzymes. Chymotrypsinogen to chymotrypsin, proelastase to elastase, procarboxypeptidase to carboxypeptidase, and proaminopeptidase to aminopeptidase. So this proteosis gets converted to peptides, and peptides are acted upon by peptidase enzyme and get converted to individual amino acids. We have 5 different transport mechanism for transportation of amino acids from the lumen into the general circulation, and this is sodium dependent active transport. For neutral amino acids, Mr. cycle is involved. With the help of reduced glutathione, neutral amino acids are transported from the lumen into the bloodstream. So coming to disorders associated digestion absorption of proteins. So wherever there is echlorhydria, there is loss of acid or decreased acid production, or if the patient has undergone gastrectomy, partial gastrectomy, removal of part of the stomach. So what happens? There is no HCl, there is no stimulation conversion of pepsinogen to pepsin, so there is impaired digestion of proteins. So the next set of disorders are associated with pancreas. It can be in the form of acute pancreatitis, where there is damage to exocrine pancreas, chronic pancreatitis, chronic inflammation, alcoholic pancreatitis, and in carcinoma of pancreas. So what happens? The exocrine pancreas is damaged and the enzymes are not active, so impairing the digestion of proteins. So the next set of disorders at the level of small intestine include the Hartman's disease, where there is defective absorption of neutral amino acids. So there will be impaired digestion absorption of proteins. Osteo syndrome is due to impaired absorption of methionine. Oxoprolinuria is due to deficiency of oxoprolinase enzyme, which is responsible for conversion of oxoprolin to glutamate. So the Meester cycle is disrupted. Then the protein losing entropathy, where there occurs loss of proteins in the intestine resulting in hypoproteinemia, and celiac disease characterized by villus atrophy, so there is no absorption of protein. So these are the different disorders that can impair the digestion absorption of proteins. Coming to what is the fate of these amino acids in well-fed state. So we all now know proteins are broken down to proteoses, then to peptides and individual amino acids, and by 5 different mechanisms these amino acids are released into bloodstreams. Once they are released into bloodstream, what is the fate of these amino acids? So brain has got special linkage for branched chain amino acids, because these can easily cross blood-brain barrier and they release less of ammonia. So brain prefers branched chain amino acids for its metabolic purpose. So glutamine is selectively taken up by kidney, where glutamine releases ammonia. This ammonia gets converted to ammonium ion, and this is responsible for maintenance of or regulation of pH, and thus it maintains acid base balance. All other amino acids are taken up by liver, and where they are utilized for protein biosynthesis by translational. Now, what is the fate of these amino acids when we are fasting or in starvation state? When we are not consuming any food, what happens is the muscle proteins are broken down. So, of these, the branched chain amino acids are selectively taken up by brain, glutamine is selectively taken up by kidney, all other amino acids go back to liver through portal vein, and here they undergo oxidative deamination, releasing ammonia, which gets detoxified to urea, and the carbon skeleton of this amino acid is used for gluconeogenesis, that is synthesis of glucose from non-carbohydrate source. So this was all about digestion absorption of proteins and its related disorders. Next video will be on formation of ammonia and urea. Thank you. Please do like, share and subscribe this video. Thank you.